Gonzalo Parra is an Argentine scientist. He got his bioinformatics degree and his PhD from Buenos Aires University in Argentina. He was a postdoctoral researcher at the Max Planck Institute for Biophysical Chemistry in Germany. And then he moved to work as a postdoctoral fellow in the German Cancer Research Center. And since 2021, Gonzalo works at the Barcelona Supercomputing Center, where he tries to understand the molecular mechanisms behind protein evolution, stability, function, and dynamics, both in health conditions and in disease. So you can start your presentation whenever you're ready, Gonzalo. Thank you.
Thank you. Thank you for the invitation and for allowing me to present here. I'm very excited. It's a different community, so let's see if I can convey my message. So my talk is titled Local Energetic Frustration Through Time and Species. But I suppose that not everybody knows what frustration means in this context, so I will explain a little bit before going to results and how to apply it to our studies. So the first thing you have to understand is that proteins are evolved systems that are not only optimized to fold. Many of proteins, they need to fold, but they are not only optimized to fold, but also to function. So to give some context, how we understand protein folding, we understand it in the context of the energy landscapes theory that says that proteins are very specific systems that are selected by evolution in such a way that they fulfill the principle of minimum frustration, which means that when you look at the landscape of a protein, this is the unfolded state, this is the folded state. When you go to the folded state or native state, most of the interactions that are present in this structure are good for the stability of the fold, right? But whenever you look more carefully at the native state of a protein, you can always find that some specific interactions remain in conflict, in energetic conflict with its local surroundings. And we call these interactions highly frustrated because it doesn't matter what the protein does, it cannot minimize energetically all the interactions at the same time. So why do we have these highly frustrated interactions is the question that Peter Wallness and my PhD supervisor tried to answer many years ago. Well, not so many years ago. So the main answer that they found to be feasible for this to happen is that if you look at the native state of a protein, it is a collection of conformations that exist in a dynamic equilibrium, and these local frustrated interactions or local conflicts are the ones that allow the protein to move in between the different conformers that constitute the native state and finally give rise to the functional features of a protein. So later on, Diego Ferreiro, who was my PhD supervisor, developed an algorithm that is named frustatometer that allows us to find, localize, and quantify frustration in protein structures. So the algorithm is very simple. Once you have a protein structure from a PDB, what we do is to define which residues are in contact with each other. Once we have this collection of contacts in the protein, we use an energy function to measure what's the energy between two given amino acids in the structure. We calculate with this energy function what's the native energy, and then we generate decoys, or what is also named perturbations of the structure. We do this 2,000 times because this is a number that works well, and for every of these decoys, we calculate again what would be the energy in that alternative structure that we generate. And with these 200 decoys, we can generate a distribution of energies against which we are going to compare our native energy. So basically what we do here, for you to understand it very easily, is if I mutate my structure 2,000 times, and most of the times the energy is worse when I mutate, it means that the energy in that point was optimized or that it was minimally frustrated. On the contrary, if most of the times I mutate the structure and the sequence, I get a better energy, it means that the energy in that specific loci was not optimized, and then we consider that it is highly frustrated. If the mean energy of the distribution is very similar to the native energy, then it's a neutral. It's not there, not so bad, not so good. That's the main thing you have to remember for the rest of my talk. So later on, when I started my PhD, we worked a lot to get first a web server where you can easily input your PDV structure, and then you can get a frustration measurement either for all the contacts in the structure, and it will look like this. Red is highly frustrated, green is minimally frustrated, and neutral interactions are not shown because there are so many and they difficult visualization. But also you can get a frustration measurement for each residue, so you can have it either for contacts or for residues. And more recently we have also developed an R package which allows you to do this locally in your computer, to put it in a supercomputer in a cluster, and then you can calculate frustration for thousands and thousands of proteins. And we as well, we are advancing to do this large scale in what we now call frustatomics. So how do we interpret frustration residues? As I said, the only thing you have to remember is this. Once you have a protein, you can classify each residue or contact into being minimally frustrated, which is the native energy is statistically more favorable than random variance; highly frustrated, which means that the native energy is statistically less favorable than random variance; or neutral in the middle case. So when we apply this to a statistically representative set of structures in the PDB, what these people observed in 2007 was that up to 10% of all the interactions that are present in the native state of a normal globular well-behaved folded protein are in conflict with its local structure. 40% of these interactions are minimally frustrated, which means that they contribute positively to the stability. They are mostly located in the hydrophobic core, and the rest is neutral. These highly frustrated interactions, they are not located randomly in the structure, but they are enriched in certain parts of the proteins. In different studies over the years, they found that these 10% of interactions are specifically enriched in protein-protein binding sites, allosteric sites. More recently, we checked that they are also quite enriched in active sites. But the thing up to here, and you can go and read these papers, is that up to here we were measuring frustration as being enriched in big data sets. But these big data sets are heterogeneous, because if you take all the binding sites together and you measure statistically how enriched they are, you find that the highly frustrated interactions are enriched. But what happens if you want to go to a specific binding site in a specific protein, or allosteric site or active site in a specific protein? It's not so easy, because then the measurements are not so reliable when you study frustration in a single protein structure. So how we did to overcome this issue is to use evolutionary information. So the questions now are, can we use evolution to better understand frustration? And can we use frustration to better understand evolution? And I will also spoiler you a bit here, because in my talk, I will not talk about the effects of frustration invariance. I am going to get to the end where I will pose how we can use this to understand protein variants, because this is a project of one of the people who are working with me, who is starting the PhD and is going to bring this further towards that dimension. So mainly this evolutionary dimension of frustration was developed by Maria Freiberger and Victoria Ruiz Serra, and it goes like this. Let's suppose we have a protein from humans, and we calculate frustration. We classify all the residues into minimally frustrated, neutral, and highly frustrated. If we take this residue here that is red, we say that usually this correlates with functional information, but it's very difficult to know if this is important or not. So one way to overcome this challenge is, if we have this protein and we transfer the colors from the structure to the sequence, we have this sequence colored by frustration states. And then we can do the same for homologous proteins in other species. We can do this, for example, same protein, the homologous in the cats, in horses and dogs, and we just transfer the colors from the structures to the multiple sequence alignment of all the proteins. And then once we do this, we can see some patterns. We see that this color here that is red, this tyrosine that is highly frustrated, is not only highly frustrated in the human protein, it's also highly frustrated in a big data set of homologous proteins. So the possibility that this is here highly frustrated because of functional reasons is very high. Why do I say because of functional reasons? Because if this conflict wouldn't be desired, then evolution would have minimized the energetics of this loci here. You can see that some other regions are minimally frustrated, like this one here, even when the sequence can change. So this could be whatever hydrophobic residue, but other regions are not so conserved. For example, here you have a lysine, but the state changes in between proteins. So the energetics is much more, it can be fine-tuned in a much more fine-grained way than sequence. So in order to be able to evaluate this in a quantitative way, we have seen that you have regions with low frustration conservation, some other regions have high frustration conservation, and in order to see how this relates or not with sequence, we can calculate what we take from the information theory concepts like information content, and given the distribution of sequence symbols or colors in each of the columns of this multiple sequence alignment, we can quantitatively measure how much conservation we have of each of the symbols in each of the columns of the multiple sequence alignment. And then you can have things like this. I guess most of you know what a sequence logo looks like, which is like this. So for example, in this part that has low conservation, you see that this position here is not very well conserved. And you can calculate the same for frustration states, and you see that here is not well conserved. Whereas in this part where everything is highly conserved, you can see that the sequence is very conserved and the frustration color is also very well conserved. So how do we use this for meaningful studies in protein families, for example? So what you need to understand is that when the color is conserved, green stuff are good for stability. Red stuff are bad for stability, and if something is bad for stability and conserved in a great majority of protein members of the same protein family, this points out that this conflict might be because of functional reasons. So the first time we applied this to a protein family, we did it some years ago, like 2018, to the beta-lactamases family. We took 31 experimental structures non-redundant from this family, and we performed our calculations with the structures and the multiple sequence alignment. And what we observed is here in asterisks, you can see the catalytic sites of a protein, and you see that there are many of these residues that are highly frustrated and conserved. So this means that the catalytic sites are in conflict with the local structure of the protein. And the explanation behind this, it is that the structure gets stabilized when the substrate binds. So this is a thermodynamically driven process, because when you do not have the substrate, there is a conflict. When the substrate binds, the conflict is compensated. But not only catalytic sites were shown to be in conflict with the local surroundings, also some other residues like this one here or this one here that were related to other functions. So that's when we started to believe that this local conservation of local frustration could be used in a more general way. So can local frustration conservation be a more general tool? That we'll try to convince you in the next sets of slides. So basically, the first paper where frustration was shown to be applicable in a large-scale scenario was this one from 2007 from my PhD supervisor. But then there was an accompanying paper where they analyzed which were the consequences of these frustration signals in a very specific protein, which is called IM7 protein. This protein is present in bacteria. It's 87 amino acids long single domain protein that its function is to inhibit another protein which is called colicin E7. If you look at the structure of this protein, you can see it is like this. Again, green is stability, red means something that is not good for stability, and we observe that the region where we know there is a binding site between IM7 and E7, which is not present here in this drawing, we can see that these residues here, these two tyrosines, are involved in many highly frustrated interactions. The Warnings group, what they did is to study computationally what's the folding mechanism of this protein. This is the unfolded state, this is the native state, and they observed that this frustration here leads the protein to have an intermediate state that is visible through computational techniques, but also by experiments. And this intermediate here is the direct consequence of having this frustration in this binding site. But you know that having intermediates, having local frustration binding sites that are exposed is kind of complicated or problematic in the cellular medium. And we know from later studies that the mechanisms that frustration have, that evolution have found in order to compensate for these problematic regions, because this could lead to misfolding or aggregation, is, for example, the usage of chaperones. Chaperones, we know now that they recognize these highly frustrated patches and then they bind to these proteins and only release them when they are ready to bind to their partners. So this is a very nice story from almost 10 years ago, no, almost 20 years ago. And the question is it true that evolution can help us to understand this system better? So what we did is to look for homologs of this protein, and we predicted the structures with AlphaFold, and we calculated our frustration patterns in the entire dataset. So we have this dataset here. You can see that here you can find the two tyrosines that were present here in the binding site, and they're responsible for the interactions with colicin E7. So you can see in the whole set, these residues are conserved and highly frustrated. But something that was not reported by the time was that there is another residue which is highly frustrated in the entire set, almost in the entire set, except in this protein here, and which is this tryptophan that also appears here. So when we were checking bibliography, we observed that this residue is actually important for stabilizing the folding intermediate I just described before. So this is a very interesting finding, because I told you that the intermediate state was a consequence, an undesired consequence of the frustration that we need to have in the binding site, which is localized in these two tyrosines here. But if we have this local frustration in the native state, and this local frustration in the native state stabilizes the intermediate state, which is supposedly not desired, this makes no sense. So the hypothesis we post in this paper, which was published this year, is that first, you can use frustration conservation to unveil overseen residues that were not detected as functional before, like this tryptophan here, but also to make novel hypotheses, because if this amino acid here is highly frustrated in native state and is stabilizing an intermediate state, maybe the intermediate state is functional itself. This is a question we pose. We haven't answered it yet, but we just put it there just to showcase the possibility of establishing new hypotheses by using local frustration. So one thing I want to continue showing here is how we use local frustration conservation to understand diversion patterns in between evolutionary related proteins. So one of my favorite examples is hemoglobin, because it's composed of two pairs of alpha globins and beta globins. These two proteins belong to evolutionary related families, so we know they have a common ancestor. When we calculate our frustration conservation patterns mixing the two families, we do not observe many good things, except that what it is conserved is whatever is important for stability that we see here in green. In black, we see things that are not conserved evolutionarily speaking, and only this thing here is highly frustrated, but there is not any functional relevant thing reported in this position. But when we separate our two families, we can observe, this is the dataset with the two families together, and this is the alpha and this is the beta globin. we can observe that different positions are read in differentially in between the two protein families. And because these two families are homologous, we know, for example, that this position here was the same in the ancestor. We don't know what was first, if the lysine or the glutamine, but we know that the lysine here is highly frustrated and conserved, which points out at a functional requirement, whereas the glutamine is not. So by checking bibliography, we check that all the positions that are highly frustrated and conserved, they are related to functional constraints like protein-protein interactions, interactions with a chaperone, and salt bridges. And the question is, can we go further? So this method allows us to, when we have evolutionary related families, to analyze them and to point out at what it's known in the field as specificity defining positions. But the question is, can we give an answer to what was first? Can we understand which are the mechanisms of evolutionary adaptation towards differential functions in related protein families? So this is the work that Marco Ludaik has started in our group since some time ago. So he made use of this paper from some years ago, where these authors were studying the origin of complexity in hemoglobin. So basically they have a phylogenetic tree, and then they made ancestral sequence reconstruction of some of the members in here. So what Marco, and these sequences were reported. So now that we have AlphaFold, what Marco did is to predict the structures of these ancestors and to measure frustration. So here we have pseudotime, which is how far in the past, in terms of millions of years, these sequences are. And then here we have extant globin families. We added neuroglobin and myoglobin, and these are separated just for visualization reasons. So we can see first the ancestral residue in these proteins was the lysine, which is highly frustrated, so the functional requirement is much more ancestral than the adaptation towards less frustration things, like in beta globins, where the conflict got resolved to go into a less frustration state with the glutamine. And this is because globins, they take the oxygen in the lungs with interactions with alpha globin, which has a higher conflict, and then it's released in the blood by interactions with beta globins. And neuro and myoglobins on the other side, these are monomeric proteins, so they have completely reduced the conflicts because they don't need them anymore. So this is a good example how we can check how the evolutionary trajectories in terms of frustration have evolved. So what Marco has done after is to reconstruct all the internal nodes in this phylogenetic tree. So we have now, for each internal node here, we have a sequence, we have a predictive AlphaFold structure, and for every residue in the multiple sequence alignment, we have this time resolved trajectory of how frustration changed from the ancestor towards the leaves. It doesn't matter much, but what we do now is, okay, we have this position that I have shown here, right? This position. How did changes happen? So I told Marco, why don't you go from the ancestors to the leaves and you check, this is the position that we like, you check what in a given trajectory, which changes have happened if you walk in this tree through the ancestors towards the human version or to the human version of beta globins. And you can see that for this site, different changes have happened over time. Change in frustration, yes. The alpha globin never changed from the ancestor towards today. It never changed the identity, but then other changes happened. Whereas beta globin, we know that at this moment in time, there was a change, and then some changes happened before. So we could start thinking of this to be epistatic changes that were necessary to remodel this position here in order to the protein to be able to further evolve towards the function that it has today. And then perhaps these changes that we observe after the main change happened in the specific functional position, they are just fine-tuning changes that happened towards extant times. Of course, this is a work in process. We need to finish it, but we are towards having a a computation method to understand how evolution happens in terms of energetic adaptations towards function and stability. So, concluding remarks, I think I'm almost out of time, is that frustration patterns can unveil stability and functional constraints in protein families. At higher levels, when we mix too many things together, we can retrieve stability anchors while functional signals are averaged out. This is, for example, when you mix alpha and beta globins. But when you separate evolutionary lineages like alpha and beta on their own, you can retrieve functional signals that you can assign to known functional patterns. And we are developing a novel strategy to track trajectory changes that constitute family-specific functional annotations. So lastly, I want to thank to all the collaborators during these years that have helped us to develop these tools, and I'm very happy to take questions if there is any. Thank you very much.
Thank you, Gonzalo. Thank you for your very wonderful presentation. I have two questions mainly, and if someone else have a question, please write it down in the chat or in the QA part of the Zoom. So I was thinking, what's the correlation between frustration and clinical significance for reported variants?
Yeah. Do you still see my
Yes.
Yeah. So I didn't have time, I mean, to tell everything, but just to give you an idea. So you may know this paper because you are involved in this group. So for example, you can measure experimentally what happens if you mutate every residue in a given protein by whatever other variant you may have in the alternative amino acids, right? And in our case, we correlated stability with the tallness of our bars, and we see that there is a correlation between what is good for stability and the experimental stability that you can measure by these full deep scanning experiments. In particular, for example, what is interesting that this other paper came out later, and just the summary of this is that there is a correlation for things. So the green dots here, it means that if you mutate those positions, we predict that the stability is going to be decreased, which is in correlation with what you can measure experimentally. But this position here, for example, which is this position here, which is important to bind the substrate, this is the KRAS protein. So this position here that is important to bind the substrate, it goes the other way around. This point here with this experiment, it means that most of the variants you make here, they are better in terms of local stabilities, but you are messing up with the function basically. So the ultimate consequence with this is that you may have a pathogenic variant if you touch this position here. So if you followed my logic is, if you have a sequence of a protein of interest for you, let's suppose a cancer scanning in a panel, you know you have a clinical variant that you know what's doing there. You can get the homologous proteins of this, you can predict the structures, and quickly this takes maybe all and on, you can have this in half a day. So you can see if your variant falls either in a evolutionary constraint and green position, which means that your variant it's affecting the stability of the protein, so it could lead to misfolding, it could lead to, I don't know, you know better than me, better what this could happen. Or if this is a functionally constrained position, like it's red and conserved, which is telling you that the variant here might be pathogenic because it's conflicting with the function itself of the protein. I think that maybe answers your question, and this is something we want to tackle in the immediate future because we're going to apply this to all proteins that are related in cancer databases, for example, SNPs related to cancer, and want to classify these patterns in terms of benign or pathogenic driver mutations and so on.
Okay, and then I have another question, because you pointed out that some positions may be frustrated, for example, in human, but not in the homologous sequences. And we know that in many cases we tried drugs in mouse, they cured a certain disease, but then when this drug moved to the clinic, it's not working in humans. So could these positions where frustration is not conserved be pointing positions that are behaving different in human than in other species? Like certain functionality that is relevant for the human protein, but not in other?
Not really. I don't think so. I would love that this would be possible, but I think the problem with frustration in single proteins is that you never know if this is relevant or not. So
Mhm.
you have two strategies to give relevance to these signals. Either you add the evolutionary dimension, as I did here in this Frustra Evo approach, or you add the dynamic nature of the native state, which means you make an equilibrium molecular dynamics and you see how frustration changes across the different conformers of your protein, so you can have a better sense of how relevant this frustration signal is in the native ensemble of the protein. And of course, in between humans and mouse, it could be also that the complete interaction network, protein-protein interaction networks, is different. So I don't think you can get an answer by looking at differences in between specific species. You can better understand things that are like this. For example, this case, let's suppose you have human and mouse here. It means, and many other homologs, it means that in mammals, for example, this position is very similar in terms of sequence and frustration. And then you can be confident that the effect of this variant is affecting something that you don't want to affect. But if you have one protein that deviates from the ensemble, let's suppose one of these proteins doesn't have a lysine here, interpreting what that change means, at least with the tools we have developed so far, I don't think it's possible.
Okay, thank you. And then we have some questions from the audience. Didier De Bois is asking, when you said that the frustration of a highly frustrated residues in binding sites could be reduced upon binding, is this mostly a hypothesis, or is it something that you quantify by calculating frustration in the complex using the method you presented at the beginning with the cause,
Yeah.
etc.?
Thank you for the question. So here, if you go and read this paper, they measured basically frustration is conformation dependent. So if you have two conformations of the same protein, it gives you two different frustration values because it depends on the conformation. If you measure, let's suppose you have hemoglobin, right, which has protein-protein interaction sites. If you measure frustration on alpha globin alone, you expect to see highly frustrated interactions in the protein binding sites because they are not going to be compensated by the interacting partners. If you give to our algorithm the entire quaternary structure, then if you go and look in those sites, these are going to be minimally frustrated because now the interactions are compensated. And basically this is what drives the process in thermodynamic terms because the process is favorable. So it depends on what you want to see. And also there is another paper from 2015 where we studied the entire family of ankyrin repeat proteins, which are protein-protein interactors, where we repeated this analysis, and we have also some other conclusions from that work. So yeah, it's you have different results depending on what you are observing and giving as input, and your interpretation needs to be accommodated accordingly.
Okay, thank you. And then the last question, Seven Diderich is asking which of the tools would you now recommend to calculate frustration per ratio for a given structure of interest?
Oh, you can use, if you only have one protein and you want to check how to run it, how the output looks like, I recommend to go to the web server. If you have a protein, that is not extremely big, let's say more than 2,000 residues or so, the results are in a couple of minutes. And then you can quickly see if this is what it's useful for you. If you consider that the algorithm is useful and you want to apply it large scale, then you can try to install the R package. But there's always dependencies and environments, and this is not so easy for somebody who is not having some computational skills. But of course, you can always write to us, and we are going to be very happy to help you. So I recommend to start with the web server, and if you find it useful, try the R package, and if you find it useful and cannot install it, just write to us.
Okay, thank you very much for your presentation again. Thank you, Anna, as well. Well, this is all for today, and we will be looking for you for next seminar on June. So thank you very much.
Thank you. Bye.